Chapter 5 covers genetics. So genetics is the study of genes, and genomes are the entire collection of genetic material in a cell or a virus. So cells have DNA, and they also have RNA. Now, one interesting thing that occurs out there in nature, guys, are what are known as plasmids. And plasmids are pieces of DNA that exist outside of the chromosome. And it's interesting because some of these are related to antibiotic resistance, and that's something we'll talk about later in the semester. Now, nucleic acids are built from what are known as nucleotides. So please know that nucleotides have three basic parts: a phosphate group, a sugar, and a nitrogenous base. In DNA, the four bases are adenine, guanine, cytosine, and thymine. A will always bond with T, and G will always bond with C. Now, DNA is a double helix, so as you can see from this picture here, it's twisted around. And then these bluish lines in the middle, those refer to the bonds between the different bases. So again, you will have your sugar phosphate backbone, then your bases are in the middle here, again, A and T bond, G and C bond, and so on. So they're considered complementary in that respect. Now, another feature of DNA is that it has what's called an anti-parallel arrangement. So what that means is that the 5' end of one strand lines up with the 3' end of the other strand. RNA is different. It's ribonucleic acid, and it also has a phosphate group. The sugar is ribose, but we have one different base. It does have adenine, guanine, and cytosine, but instead of the thymine, we actually have uracil instead. So please know for your exam that the three types of RNA are messenger, transfer, and ribosomal RNA. Now, we're going to discuss DNA replication. You'll have to know the steps of this. And these are the main enzymes that are involved. So as I walk through the steps, I'll discuss the different enzymes that you would need to know for the exam. So this is just a cartoon, and on the next slide I will walk you through the steps and explain them in a little bit more detail. But first you have to unwind the DNA, then you have to break the bonds, then you have to start copying your DNA, and then eventually on what's called the lagging strand, you've got to stick them together because little fragments get made. So let's walk through what you need to know. So in step number 1, an enzyme called topoisomerase unwinds the DNA. Then the enzyme helicase breaks the hydrogen bonds between the two DNA strands. Single-stranded binding proteins keep the strands apart, and that's very important because anytime A sees T or G sees C, they want to come back together. So there's two strands. So the strand called the leading strand, let's talk about that first. On the leading strand, the DNA polymerase will bind to a primer and continuously copy DNA 5' to 3'. The other strand is called the lagging strand. On the lagging strand, DNA polymerase binds to several primers and copies DNA 5' to 3' in short fragments. Now those fragments have to be put together. So in step 6, DNA ligase seals the fragments together. Now, DNA replication is considered semi-conservative. So what that means, if we start out with one cell and the DNA gets replicated, each daughter cell will get one new strand of DNA and one old strand of DNA. So again, if you start out with one cell and the DNA gets replicated, each daughter cell gets one new strand of DNA and one old strand of DNA. Now we'll be talking about protein synthesis as well, and that's basically when you're making proteins. That's what it means. And you're going to have to first copy some RNA, and that's called transcription. And then when you use the RNA to make proteins, we call that translation. Now another interesting factor, guys, that can occur is that you can actually take DNA and go back to RNA. Usually it's DNA to RNA to proteins, but sometimes DNA is copied back into RNA. So please know that if you're going to make DNA from RNA, we use the enzyme reverse transcriptase. Again, reverse transcriptase will make DNA from RNA. And again, for your exam, please know messenger RNA that contains the codons, transfer RNA, anticodon loop. The ribosomal RNA folds into this really cool big 3D structure that will help bring the proteins and amino acids together so that you can make your protein. Now, one feature of eukaryotes is that we have a lot of spacer DNA, I'll call that. So for your exam, please know that introns are non-gene regions. So they will actually separate the gene regions, and the gene regions are referred to as exons. So when RNA is being processed, introns get spliced out before you actually have the messenger RNA that you need to make your proteins. All you need to know is exons are the gene regions and introns are non-gene regions. And here again is an example of what the ribosomes look like. Now this is our genetic code chart that will show you the different types of codes needed to bring in amino acids. For your exam, you'll have to know the start codon is AUG. Again, start is AUG. And also know that we have 3 stop codons. You don't have to know the code, but know we have 3 stop codons. You'll have to know the steps of protein synthesis as well. So let's go through those. So the ribosomes come together at that start codon AUG. The first amino acid is methionine, and that's brought into the P site by transfer RNA. The next amino acid is brought into the A site by transfer RNA. A protein bond then forms between the amino acids. The transfer RNA then that's in the P site leaves through the E site, and the ribosome moves forward 1 codon. So we repeat the steps 3 through 6 until you reach a stop codon. Now bacteria are interesting because they have to replicate relatively quickly, and they have what are known as operons. So please know, operons are a collection of genes controlled by a shared regulatory element. Now, many times in the environment, bacteria have to communicate with each other. So please know that the communication between the bacteria is called quorum sensing. Again, quorum sensing is the communication between bacteria. Mutations are something we need to be aware of, and the 3 that I'd like you to know for your exam are substitution mutations. They occur when an incorrect nucleotide is added. Insertion mutations. These occur when a cell adds 1 or more nucleotides to its genome sequence. And then deletion mutations occur when 1 or more nucleotide is removed from a genome sequence. So please know these 3 types of mutations for your exam. And lastly, please take a screenshot of this slide, guys. We have what are known as transposons, and transposons are jumping genes. So what happens is that those jumping genes can be inserted into DNA, and some interesting facts with that is you might actually wind up with the gene for a toxin, for instance, in a bacteria, or you might wind up with resistance to an antibiotic. So these transposons, or jumping genes, are very important.